Welcome everyone. Today we're going to be starting our exploration into the lymphatic system. This system may not get as much attention as the circulatory or the nervous system, but it actually plays a vital role in our overall health and immune defense. The lymphatic system is essentially the body's drainage and surveillance system. It helps maintain fluid balance, transports fats from the digestive system, and plays a central role in our immune function. We'll be covering the structure of the lymphatic vessels, nodes, and key organs like the spleen and the thymus. We'll also introduce major components of the core function of the lymphatic system. As we go through this chapter, you'll see how it's integrated with other systems, including the circulatory system and the immune system, and why it's essential for maintaining homeostasis. These are our learning objectives for today's lecture on the lymphatic system. First, you'll be able to describe the key components, which include the lymph vessels, lymph nodes, and organs like the spleen, thymus, and tonsils. You'll learn not just where they are, but what they do and how they're built to serve their purpose. Next, we'll look at how the lymphatic system integrates with other body systems, especially the cardiovascular system and immune system. This system does not work in isolation. For example, the lymphatic vessels return excess interstitial fluid to the bloodstream, and lymph nodes are packed with immune cells that monitor for pathogens. Finally, you'll learn how to analyze the structure-function relationship, how the architecture of the lymphatic vessels allows one-way flow of lymph, and how lymph nodes are strategically positioned and organized to optimize immune surveillance. Let's begin by introducing the lymphatic system and its essential role in the body. The lymphatic system is often described as a secondary circulatory system because, like blood vessels, it forms an extensive network of tubes that permeate nearly every tissue. But unlike the circulatory system, lymphatic vessels carry one-way flow of lymph rather than blood. Lymph is a fluid that originates as interstitial fluid, the fluid that leaks out of the capillaries into the spaces between them. The lymphatic system includes this fluid and eventually returns it to the bloodstream, helping to maintain fluid balance. Without this system, we'd experience fluid buildup or edema in our tissues. Another function is in the absorption of fats. Specialized lymphatic capillaries called lacteals found in the linings of the small intestine also absorb dietary fats and transports them to the bloodstream. This function complements the digestive system. Perhaps most critical, the lymphatic system plays a major role in immune defense. As lymph travels through the lymph nodes, it is monitored by white blood cells, mainly lymphocytes and macrophages, that can recognize and respond to pathogens like bacteria, viruses, or abnormal cells. In addition to lymph nodes, there are major lymphatic organs like the spleen, which filter blood and stores immune cells, the thymus, where T-cells mature, and the tonsils, which are among the first line of defense against inhaled or ingested pathogens. The lymphatic system has several essential functions that are closely tied to both immune and the circulatory systems. One of its major roles is to support immunity by producing, developing, storing, and transporting leukocytes, which are white blood cells such as lymphocytes and macrophages. These cells are critical for identifying and fighting infections. Structurally, the lymphatic system includes a network of vessels that form a one-way system, meaning lymph always flows in one direction, towards the heart. This network collects excess interstitial fluid that leaks out of the blood capillaries and returns it back to the bloodstream, preventing fluid accumulation or swelling of the tissues. This helps maintain blood volume and fluid balance. As lymph travels through these vessels, it passes through lymph nodes. These act as filters, screening the fluid for pathogens, damaged cells, and foreign particles. The lymphatic system also contributes to digestion. In the small intestines, specialized lymphatic capillaries called lacteals collect lipids from digested foods. These lipid products are too small to enter the blood capillaries, so they are carried through the lymphatic system and eventually released into the bloodstream. The fluid itself, lymph, is similar to plasma. It contains immune cells along with nutrients, waste products, and proteins, making it a connective tissue that plays multiple supportive roles in the body. Now that we've covered the function of the lymphatic system overall, Let's take a closer look at the tissues and organs that carry out these various functions. First, we have lymphoid tissues. These are found in every single area of the body and contain fluid that is very similar to lymph. The main role is to support the life cycle of lymphocytes. They help with the production, maturation, storage, and deployment of these white blood cells. Two main types of lymphoid tissues are tonsils and MALT, or mucosal associated lymphoid tissue. MALT is located in the mucous membranes, such as the linings of the respiratory, digestive, and urinary tracts. They are critical entry points for pathogens. These tissues are composed of dense clusters of immune cells, especially leukocytes, that are embedded in the supportive reticular connective tissue framework. Their function is to help the body detect and respond to pathogens and foreign material, especially within organs of other systems. For example, the tonsils of the pharynx can help detect pathogens that enter through the mouth or nose. Then we have the lymphoid organs, which include the thymus, spleen, and red bone marrow. These organs are more structurally complex than tissues, but they serve similar immune functions. They are responsible for maintaining healthy lymphocyte populations and providing sites for immune responses to be activated. The lymphatic vessels make up a vast network rooted near the blood vessels of the cardiovascular system. These vessels play a central role in fluid recovery by collecting fluid that leaks from the blood capillaries into the tissues and returning it back to the bloodstream. This function is critical for maintaining consistent blood volume and preventing tissue swelling. Beyond fluid balance, the lymphatic vessels are also responsible for transporting dietary lipids and certain lipid-soluble vitamins from the small intestines. These nutrients are absorbed into specialized lymphatic capillaries and are carried to the bloodstream, where they can be distributed throughout the body. Another key function of the system is to distribute the distribution of leukocytes, which are white blood cells involved in immune defense and general maintenance of tissue health. These immune cells travel through the lymphatic vessels to monitor and respond to threats in various parts of the body. The fluid that travels through the vessels is known as lymph, which is composed primarily of lymphocytes and other white blood cells, as well as proteins and lipids. Lymph originates as interstitial fluid and plays a vital role in both nutrient transport and immune surveillance as it circulates through the lymphatic systems. As blood circulates through the body, blood pressure forces plasma out of the capillaries and into the surrounding tissues. Each day, about 20 liters of plasma enters the interstitial space, the area between the cells, through this process of capillary filtration. Most of that fluid, around 17 liters, is reabsorbed directly back into the blood capillaries. However, the remaining 3 liters of fluid does not return through blood vessels. Instead, this excess interstitial fluid is collected by the lymphatic capillaries, where it enters the lymphatic system and is now referred to as lymph. This continuous cycling of fluid from blood plasma to interstitial fluid to lymph is essential for maintaining proper tissue function. It ensures that cells are constantly bathed in fresh fluid rich in oxygen and nutrients, while also removing waste and potential harmful pathogens from the tissues. The lymphatic system therefore plays a vital role in both fluid balance and overall tissue health, acting as a return route for fluid that escapes the vascular system. Lymphatic capillaries, also known as terminal lymphatics, represent the starting point of the lymphatic system. These microscopic vessels are where interstitial fluid first enters and becomes lymph. They are also present in nearly every tissue of the body, particularly in the soft connective tissues. But there are a few exceptions. There are no lymphatic vessels in red bone marrow, bones, teeth, or the cornea of the eye. Structurally, lymphatic capillaries are unique. They are made up of a single layer of overlapping endothelial cells, which forms a flap-like valve. These flaps open in response to increased interstitial fluid pressure, allowing fluid to enter the capillary. When pressure inside the capillary rises, the flap closes to prevent fluid from leaking back out, ensuring one-way flow. These capillaries are held in place by collagen fibers that anchor them to the surrounding tissues. As interstitial pressure builds, the anchoring fibers pull on the endothelial flaps, increasing the space between them and facilitating fluid entry. In the small intestines, a specialized form of lymphatic capillary, called lacteals plays a critical role in nutrient reabsorption. Lacteals absorb dietary lipids and lipid-soluble vitamins that are too large to enter the bloodstream. These lipids combined with other substances to form a milky fluid called chyli, which is then transported through the lymphatic system and eventually released into the bloodstream. As lymph moves through the capillary into larger lymphatic vessels, the structures of these vessels becomes more complex. These larger lymphatic vessels are structurally similar to veins. They have 3 tunic layers in their walls and are equipped with one-way valves that prevent the backflow of lymph. These valves are spaced closely together and create visible bulges along the vessel, giving it a beaded appearance. Unlike blood in the cardiovascular system, lymph is not actually pumped by the heart. Instead, it is propelled through the vessel by external forces, including the contraction of skeletal muscle during movement and the pressure changes that occur during breathing. These forces generate a pressure gradient that pushes lymph forward, and then the valves ensure it moves in one direction. As lymph travels through these vessels, it passes through the lymph nodes where it is filtered for debris and pathogens. The human body contains approximately 500 to 600 lymph nodes, which are positioned at intervals along the lymphatic vessels. Some are more prominent clusters of lymph nodes, like the ones found in the axillary region near the armpits and the inguinal region near the groin. The lymphatic vessels themselves are organized into 2 main systems: superficial lymphatics, which run alongside veins in the skin, and deep lymphatics, which follow the path of arteries in the body's internal organs. Eventually, these vessels converge into one larger lymphatic trunk, which collects lymph from broad regions of the body. The final step in lymph transport occurs through the lymphatic ducts, which are the largest lymphatic vessels responsible for returning lymph to the venous circulation. The drainage pattern of these ducts is asymmetrical, meaning that each side of the body drains through a different route. The right lymphatic duct is much smaller and only collects lymph from the upper right portion of the body, including the right side of the head and neck, as well as the right upper limb and the right side of the thorax. This duct empties lymph into the right subclavian vein, just lateral to its junction within the right internal jugular vein. The rest of the body, eventually everything else, drains through the thoracic duct, which is significantly larger. The thoracic duct begins in the cisterna chyli, a sac-like chamber located just below the diaphragm. It collects lymph from the lower abdomen, pelvis, and lower limbs via the left and right lumbar trunks, as well as from the intestinal trunk. As it ascends, the thoracic duct also receives lymph from the left jugular, left subclavian, and left brachiomediastinal trunks. It ultimately empties into the left subclavian vein, lateral to where it joins the left internal jugular vein. This asymmetric drainage system ensures that lymph from all parts of the body is eventually returned to the blood, completing the fluid transport cycle and supporting both circulatory and immune functions. The lymphatic system also includes a wide range of specialized structures that are essential to immune function. These structures are collectively referred to as lymphoid organs and tissues. The lymphoid organs are well-defined anatomical structures that contain multiple tissue types, while lymphoid tissues are more diffusely concentrated immune cells found within the tissues of other body systems. We categorize lymphoid organs into 2, primary and secondary, which are based on their function of lymphocyte development. Primary lymphoid organs are where lymphocytes are formed and undergo development. This includes red bone marrow, where the blood cells originate, and the thymus, where T cells mature. These organs are essential for producing lymphocytes that are capable of participating in an immune response while also learning to tolerate the body's own tissues. In contrast, secondary lymphoid organs are where lymphocytes reside and become activated in response to pathogens. These include the lymph nodes and spleen and various types of mucosal-associated lymphoid tissues, or MALT. Once lymphocytes mature and leave the primary organs, they circulate to these secondary sites where they await exposure to antigens. Lymphoid tissues, meanwhile, are clusters of immune cells such as lymphocytes and macrophages that exist within other organs, like the tonsils or the small intestines. While not enclosed by a capsule-like organ, they still play a crucial role for detecting pathogens and initiating immune responses, particularly at common sites of pathogen entry. The primary lymphoid organs are responsible for generating and developing the cells that carry out the adaptive immune response. These organs include the red bone marrow and thymus glands. Within these structures, lymphocytes, specifically B and T cells, undergo critical developmental processes that allow them to function properly. These include learning to recognize foreign antigens, avoid reacting to self-antigens, and eventually remember encountering pathogens by forming memory cells. Red bone marrow is where all blood cells originate, including lymphocytes. B cells complete most of their development in the bone marrow, while T cells begin their development there but migrate to the thymus for further maturation. These developmental processes are essential. Without them, the body could not produce immunocompetent lymphocytes. These are cells capable of identifying and responding to specific pathogens. Equally important is that these cells must learn self-tolerance, meaning that they must be trained to not attack the body's own tissues. Failure in this training process leads to autoimmune diseases. Once B cells and T cells are fully developed, they leave the primary lymphoid organs and migrate to secondary lymphoid organs, where they wait to encounter antigens and mount an immune response. Red bone marrow is one of the two primary lymphoid organs that serve as the primary site for hematopoiesis, the process of blood cell formation. This includes the production of red blood cells, white blood cells, and platelets. Red bone marrow is found surrounding the trabecula of spongy bone and consists of loose arrangements of hematopoietic cells suspended in a reticular connective tissue. These developing cells are supported by a complex microenvironment that enables their differentiation. The B lymphocytes, or B cells, complete nearly all of its development in the bone marrow. This includes both proliferation and learning process, needed for immune function, such as antigen recognition and self-tolerance. T cells, in contrast, begin their development in the bone marrow, but do not complete it there. Instead, they leave the marrow as thymocytes, which are immature T cells that migrate to the thymus to undergo further specialization. It is also important to note that the role of hematopoietic organs changes over the course of the development. In the embryo, blood cells are first formed in the yolk sac, then in organs like the liver, spleen, and lymph nodes. Eventually, as development proceeds, the red bone marrow becomes the primary site for hematopoiesis in children and adults. The thymus is the second of the two primary lymphoid organs and plays a critical role in the maturation of T lymphocytes, or T cells. It is a bilobed organ located in the mediastinum, the space between the sternum and the aorta. The thymus is most active during early life, when the immune system is still developing and the body is being exposed to many new pathogens. After puberty, the thymus begins to shrink, and the tissue is gradually replaced by fibrous and adipose connective tissue, a process known as thymic involution. Immature T cells called thymocytes originate in the red bone marrow and travel through the bloodstream to the thymus. Once inside, they undergo a complex maturation process that prepares them to become naive T lymphocytes. These are cells that are fully capable of initiating the adaptive immune response but have not yet encountered an antigen. The structure of the thymus is highly specialized. It is enclosed in a fibrous capsule that extends inwards as trabecula, dividing the organ into lobules. Each lobule contains a cortex, which is densely packed with developing thymocytes and stained dark in histological sections, and the medulla, which contains fewer thymocytes and more macrophages, epithelial cells, and dendritic cells, resulting in a lighter stain. A unique feature of the thymus is the bi-thymus barrier, the blood-thymus barrier, located in the cortex. This barrier protects the immature thymocytes from being exposed to antigens in the bloodstream before they are fully developed and self-tolerant. Once thymocytes mature and enter the medulla, they are no longer protected by this barrier and can enter the circulation, ready to participate in the immune defense. Once lymphocytes have fully matured in primary lymphoid organs, They migrate to secondary lymphoid organs where they are stationed until they encounter an antigen. At this point, they are called naive lymphocytes. They are fully immunocompetent, but they still have not yet been activated by specific pathogen. The major secondary lymphoid organs include lymph nodes, the spleen, and MALT, or mucosal associated lymphoid tissue, which is found in areas frequently exposed to pathogens like the respiratory and digestive tracts. Despite differences in location and specific immune roles, these secondary organs share several key structural features. First, they are built on a framework of reticular connective tissue, which provides scaffolding for immune cells. Embedded in this framework are fixed macrophages that help capture and destroy pathogens. These organs also contain lymphoid nodules, which are clusters of immune cells with a distinct B and T cell zones. In many of these nodules, specifically in the lymph nodes and MALT, these are known as germinal centers, which are the regions where activated B cells rapidly divide and differentiate into plasma cells. The spleen is a notable exception, and this does not contain classic germinal centers, but instead it has its own organized structure of lymphocyte activation. Another shared feature is the presence of high endothelial venules. These specialized blood vessels are thicker and more columnar epithelial cells that allow for lymphocytes to exit the bloodstream and enter the lymphoid tissues directly. This facilitates constant surveillance, enabling lymphocytes to move quickly between blood, lymph, and tissue compartments. Among the secondary lymphoid organs, lymph nodes are especially important. They act as biological filters, screening lymph for pathogens, debris, and abnormal cells. They are also the primary site where adaptive immune responses are initiated. This is where T cells and B cells first encounter antigens and become activated. Lymph nodes are densely populated with immune cells, including T lymphocytes, B lymphocytes, plasma cells, macrophages, and dendritic cells. These cells work together to detect and neutralize threats before they spread throughout the body. Structurally, each lymph node is enclosed in a dense connective tissue capsule, which sends inward extensions called trabecula to divide the node into compartments. The interior of the node has two main regions: the cortex, which lies closest to the capsule and contains lymphoid nodules where B cells can proliferate and form germinal centers. The medulla near the hilum is rich in immune cells and plays a key role in filtering and processing lymph before it exits the node. Lymph enters the node through the afferent lymphatic vessels located on the convex surface inside the node. Lymph flows past dense clusters of immune cells after filtration. It exits through one of the two efferent vessels at the hilum, the concave side of the node. One-way valves in the lymphatic vessels ensure that lymph flows in the correct direction, into the node, through its compartments, and then out. Swollen nodes are common clinical signs of immune activation, often seen during infections, when the nodes are actively filtering lymph and producing immune cells in response to a pathogen. The spleen is the largest lymphatic organ in the body and plays multiple roles in immune surveillance, blood filtration, and erythrocyte turnover. It is located in the upper left abdominal quadrant just beneath the diaphragm and lateral to the stomach. Structurally, the spleen is surrounded by a dense, irregular connective tissue capsule which sends inward extensions called trabecula. These trabecula divide the spleen into compartments called nodules, each with specialized tissues. The internal anatomy of the spleen includes two functionally and structurally distinct regions: the red pulp and the white pulp. The red pulp makes up the majority of the spleen's mass. It consists of mostly erythrocytes and is responsible for phagocytosing old and damaged red blood cells. It also contains fixed and free macrophages and other blood cells. The red pulp surrounds sinusoid capillaries that allow for slow filtration of the blood. The white pulp is found surrounding arterioles and resemble lymphoid nodules. It contains a germinal center which actively divides B cells and is surrounded by a zone rich in T cell, macrophages, and dendritic cells. This is where the immune response to blood-borne pathogens are initiated. Between these two regions is the marginal zone, which serves as the transportation area. It is rich in antigen-presenting cells and is key to site to interaction between circulating antigens and lymphocytes. Through its combination of immune and hematologic functions, the spleen serves both as a blood filter and a defense lymphoid organ, uniquely positioned to respond to systemic infections. In addition to lymphoid organs, the lymphatic system includes lymphoid nodules and MALT, or mucosal-associated lymphoid tissues, which are critical for immune defense in areas of the body routinely exposed to environmental pathogens. Lymphoid nodules are small, dense clusters of lymphocytes embedded in reticular connective tissue. Unlike organs such as the spleen and the lymph nodes, nodules are not enclosed by a capsule. They are commonly found in the mucosal linings of the respiratory and digestive tracts, which are frontline regions where the body frequently encounters pathogens. Other well-known groups of lymphoid nodules is the tonsils. This includes the pharyngeal tonsils, also known as the adenoids, the palatine tonsils on either side of the oropharynx, and the lingual tonsils at the base of the tongue. Tonsils contain deep crypts, which are invaginations of the epithelial lining. These crypts trap and expose foreign material like bacteria and food particles to underlying T and B cells, helping stimulate the adaptive immune response. This exposure helps develop immunity early in life, though in some cases tonsils may become chronically infected and require removal. MALT is a broad term for lymphoid tissues associated with mucosal surfaces. It is found in the gastrointestinal tract, breast tissue, lungs, and eyes. One important aspect is the Peyer's patches of the small intestines. These are specialized lymphoid nodules contain M cells, or microfold cells, that actively sample materials from the intestinal lumen and transport it to immune cells in the nearby follicles. Together, these structures provide a localized immune response and play a central role in protecting mucosal surfaces, which are some of the most common entry points for pathogens.